Hello everyone, and welcome to our third week of the course. This will be our fifth lecture. So this is Drug Use in the Brain 3, so our continuing unit on drugs and the brain. And so far we haven't really talked much about drugs. We've really been talking about the brain and the small scale uh, processes that go on in the brain, things like action potentials, synaptic transmission at individual neurons. And today we'll actually start bringing drugs into the conversation. So, as with the rest of this unit, the overview here is we're going to look at drugs and their effects on the brain, and we started small. We looked at individual neurons, individual brain cells, and we wanted to understand how those function first, and then we're sort of building up in scale. So we moved on to multiple neurons, so the connection between neurons, which are called synapses. So we talked about that last week, and we'll come back to it some this week as well. But we sort of laid the foundation for normal neural function before moving on to what drugs do. And then really today we'll talk about the brain as a whole. So far we've talked about just a couple neurons at a time, and now we'll see how these things fit together into the entire brain. So in this session in particular, what we're going to do is look at the structure of the nervous system. And the nervous system, obviously, the brain is the biggest component of it, but the nervous system has a variety of parts, and those parts do different things. So when we talk about a particular part of the nervous system, if a drug affects that part, then we can expect those functions to be amplified or diminished. And so that's why we're going over this, is to understand the various systems that interact, so that when a drug affects one, we can understand and probably even anticipate what that drug might do. Also, we'll talk about some of the techniques behind our measurements of the nervous system. So how do we know what happens to activity in the brain or particular parts of the nervous system when a drug is taken? So we'll go over that somewhat briefly. We're not going to dwell on it. This is not a methods class. This is a class about drugs and what they do to the brain. So I'll go over how we know what we know, so to speak. But again, it's not a methods course. It's certainly not a physics course. So we're not going to get into details there. And we'll also start talking about the short-term effects that drugs can have. So next time, we'll talk about longer-term effects. But now that we've been talking about neurons, we can start talking about what drugs do, how they affect neurons and neural function. As a side note, usually I will go over the previous week's discussion questions in the first lecture of the week. This week I will actually do it in the second lecture, just to balance out the length of the two lectures. This lecture we have more to cover. Second lecture has a little bit less, so I'll talk about discussion questions in that one. Okay, so again, so far we've talked about individual neurons and how they work, how they transmit information from one end of the neuron, usually the dendrites, down the axon to the axon terminals. And of course there, the axon terminals will connect to the dendrites of another neuron, and we call that connection a synapse. So we can talk about synaptic transmission and transmission of information from one neuron to the next neuron. And today we'll start talking about systems of neurons. So these are large collections of large numbers of neurons that function together for a single purpose. They, for example, increase excitation of the heart and breathing in the case of the sympathetic nervous system, which we'll talk about in a minute, or they process rewards, or they process visual stimuli. So these neurons link together to process fairly complex types of information and produce fairly complex responses. So it takes a lot of neurons to get that complexity, to get that wide range of functions. And we'll talk about some of those systems and how the brain is organized. And then that'll be our main topic for today. So for example, the nervous system, we talk about in general, is actually comprised of a series of parts. So the biggest distinction is between the central nervous system, or CNS, which is comprised of the brain and the spinal cord. So what makes that distinction isn't really important. There's a membrane that covers those two things. Anything outside that membrane is what's called peripheral, and anything inside it is called central. But that distinction is not important for our purposes. This is just what we mean when we say central nervous system. We mean the brain and the spinal cord, and more often than not, really just mean the brain. The spinal cord does some processing, processes things like reflexes, but doesn't do a lot of intense information processing. And so usually in our discussion of the central nervous system, we'll be talking primarily about the brain. There's also the peripheral nervous system, or PNS, and it actually has two parts of its own. There's the somatic nervous system, and that is what we think of as being sort of under voluntary control. So the somatic nervous system takes in sensory input, things like sense of touch, technically not vision, but that's, again, not an important distinction. Things like hearing, anything that conducts sensory information from the rest of our body to the central nervous system is part of the somatic nervous system. And there's the autonomic nervous system. And this system controls autonomic functions, so functions that aren't voluntary, they're automatic. So things like breathing and heart rate, digestion. So these things, it's not to say that these things don't change over time, obviously they do, but they're not under our control. And while some of them are muscles, things like the heart and the muscles of the digestive system, again, they're not under our voluntary control the way that our skeletal muscles are. So we make that distinction between the somatic and the autonomic nervous systems as a way of dividing between things we can control and things we can't. So a quick word about subdivisions of the brain. How do we navigate when we're talking about the brain? So the brain has a variety of parts. And some quick terminology. So this diagram's a little busy, I realize. But once you realize a lot of the same terms get used over and over again, then it's not so bad. So for example, the brain is broken into 4 lobes. Now they're interconnected. I don't mean broken in the sense of separate physically. They're broken down that way for the convenience of geography. So there are 4 lobes. There's the frontal lobe, shown here on the left. In this diagram, obviously you can see the outline of the person's nose and lips. So they're facing to the left. So we're having this is a side view of the brain. So we're looking at the left surface of the person's brain, as though their skull had been pulled off. So we have the frontal lobe, shown here on the left. And the frontal lobe is really the part of the brain that's biggest, relatively speaking, in human beings. And it's thought that the frontal lobe is responsible for our complex decision-making capabilities, our what's called executive function, the ability to override instincts and impulses, and higher reasoning. So that's the frontal lobe. So naturally, if you get an injury in the frontal lobe, you start to lose these functions. Now, you don't necessarily lose all of them, but injuries to the frontal lobe usually result in an inability to control emotions, an inability to reason. And so the frontal lobe is obviously important for our day-to-day lives. In fact, all the lobes are important. But the frontal lobe is one of the things that seems to make us human. There's also the parietal lobe, so moving to the right. So that is it's further toward the back of the head, but still on top. And the parietal lobe does a variety of things. It's sometimes called association cortex. And it's involved in things like memory, but also in really coordinating sensory, motor information, combining sensory information. So it does a variety of things. It's not quite as specific as the other lobes are. Moving further back, you have the occipital lobe, which is the one on the furthest right in this diagram. And the occipital lobe is really about vision. So our visual system is very complex, very sophisticated. We actually have really pretty good vision. Compare the amount of our brain that's devoted to vision in the occipital lobe to something like the olfactory bulb, which is devoted to our sense of smell. Our sense of smell is really pretty pathetic, and our brain doesn't devote much neural hardware to it. but devotes a lot of neural hardware to vision. So sort of the back quarter of the brain is all about visual processing. Moving clockwise down to the temporal lobe, and this is part of the brain. There are two temporal lobes, one on each side, and they sort of wrap around the sides of the brain. And they're involved in a couple of functions. They're involved in hearing, uh, also involved in our sense of taste, and also involved in some social judgments. Now, social judgments also occur in the frontal lobe, uh, but things like uh, attributing intentions and goals to other social entities seems to be dependent on the temporal lobe. So we have these four lobes, and each one does uh, a certain set of jobs. Uh, and again, there are four of them. What there are more of are what are called gyri. The singular is gyrus. And the gyri are the bumps that you see all around the brain. The brain looks uh, lumpy, right? It's got this sort of convoluted surface. And it has that in order to increase the surface area. So the outer layer of the brain is what's called gray matter. That's where it's thought most of the information processing happens. And so to increase the amount of gray matter, the skull is only so big. So you have a limited volume that you can work with. So to increase that surface area, you can't make the whole thing bigger. What you have to do is wrinkle the surface, so you get more surface area out of the same volume. So the gyri are the peaks, so to speak. And then the sulci, singular sulcus, are the valleys. So if you hear that term, the central sulcus, the longitudinal, well, that one's usually called fissure, but fissure and sulcus really, for our purposes, mean the same thing. They're both indentations in the surface of the brain. So we have these terms, and these are the outer surface of the brain. And we actually won't be talking that much about the outer surface of the brain, mainly because a lot of the action in terms of drugs happens in the inner parts. We're just looking at the outside here. So later on, we'll look at the inner parts. Now, with that in mind, the occipital lobe we will mention at some point because the hallucinogens, which generate visual stimuli, of course have activity in the occipital lobe where visual processing happens. So we'll cross that bridge when we come to it. But this is just to give you some navigational coordinates. So when something is closer to the frontal lobe, we know what we're talking about. Okay, so we also have in the nervous system, we have the sympathetic nervous system. I'm going to put this diagram here. This is a fairly busy diagram, but don't be nervous. The sympathetic nervous system is responsible for sort of exciting the body, getting the body ready for action, initiating what's often called the fight or flight response. So this is going to do things like open up the airways so we can get more oxygen into the lungs and into the blood. It increases heart rate, but it's not all about increasing activity. It actually inhibits things like digestion and the movement of the intestines. So again, in the context of fight or flight, if you were faced with a predator or some threat, you either have to be ready to engage in combat or you have to run away from it. And so both those things take priority over digesting your most recent meal. And so what your sympathetic nervous system does is increase things you need in order to be physically active, and then shuts off things like digestion, which aren't the top priority at that moment. And of course, the opposite system is the parasympathetic nervous system, which for the most part does the opposite of the sympathetic. It slows down your breathing and your heart rate. It activates the digestive system again, and it's sometimes called rest and digest. So you have fight or flight, and you have rest and digest. So again, the parasympathetic nervous system operates in opposition to the sympathetic, and the sympathetic nervous system is largely involved with the neurotransmitter norepinephrine, which we talked about last time. And so what we'll see in later units is that drugs that tend to increase alertness, things like amphetamines, also tend to activate the sympathetic nervous system because they both use norepinephrine, that is the system in the brain that controls alertness and attention is dependent on norepinephrine, just like the sympathetic nervous system is. So you can't get that increase in alertness in the brain without also getting an increase in heart rate and a shutdown in the digestive system. So again, we have these two opposing systems, but they both depend on neurotransmitters. And we'll see, as we learn about different drugs, why that's important. Okay, so as I mentioned, we talked about the surface of the brain, but there are some important structures inside the brain that you can't really see just looking at the surface. So if you sort of peeled away the surface of the brain, or in this diagram, if you made the surface of the brain transparent, you would see those inner structures. And these inner structures are, evolutionarily speaking, older. So as I mentioned, the frontal cortex is very big in humans, but if you go far back enough, these distinctions in the cortex of the brain, the outer portion of the brain, don't really exist to the same degree in things like fish. But you do have these older structures. So the basal ganglia is a term that describes a collection of structures in the brain. Things like the caudate, the globus pallidus, the putamen. The amygdala is connected to the system, but isn't technically speaking part of it. The substantia nigra is very important for the basal ganglia. Substantia nigra is what produces dopamine. It's one of the only couple structures that produce dopamine, which is why if neurons in that structure die, you get Parkinson's disease. And what does that mean? Well, Parkinson's disease, again, is the result of death of neurons, of dopamine neurons in the substantia nigra. And once you lose those neurons, you lose some of the function of the basal ganglia. And the basal ganglia are important for controlling movements. And that's one of the main symptoms of Parkinson's disease, is difficulty initiating and controlling movements. And that's because you've lost the dopamine neurons that go into the basal ganglia and activate motor systems. They're also important for reward processing, the basal ganglia. So we have both of these functions being not entirely handled. There are other systems involved as well, but being primarily handled at a low level by the basal ganglia. So these are two what seem to be very different functions, and one of your discussion questions this week will be probing this relationship. Why is it the system does both of these things? Okay, there's also the limbic system, which again, evolutionarily is older. The basal ganglia, no one questions whether this is a real system. The limbic system is thought by many to be sort of a loose collection of different organs in the brain. So is it really a system? Well, it does seem to accomplish a certain set of functions, but the connections between the different parts of the limbic system aren't as clear. So it's a term that's fallen somewhat out of favor, but we'll use it for now. And so this has, again, a variety of components. Things like the amygdala, which is a part of the brain that you may have heard of. It's highly involved in processing fear. It's also involved in conditioning. So when we learn a relationship between stimulus and a response, the amygdala is often highly involved. Also, there's the hippocampus, which is very important for memory. And so these are the two things that the limbic system really does, is process emotions and handle memory. And so these are obviously two very important functions. And again, it's not immediately obvious why the same system would handle these two functions. But a lot of drugs will work on these two systems, the basal ganglia and the limbic system, to various effects. As we'll see, the hippocampus is very involved in memory, and it depends on acetylcholine, which is another one of the neurotransmitters we talked about last time. And so if you have Alzheimer's, what you will often take as a drug is a substance that effectively increases the effects of acetylcholine and stimulates memory. And so that works through this limbic system. So far we looked at cartoons, so if you're interested in the real thing, here are some real brain images. These are what these things really look like. And you can see a lot of the same structures that we just talked about shown here. So on the left of this diagram, you have the blue line and the red line, and these just show you the plane of what's called the plane of section, which is just, if you slice the brain this way, this is the view you get. So the blue line, if you cut the brain along that line and look down, take off the top part and look down, what you'll see is something like the middle panel. And so the front of the brain is on the left, the back of the brain is on the right, and you can see the basal ganglia. So they're those sort of kidney-shaped structure in the middle. So you can see the caudate, which is right here, and you can see the putamen, and you can see, for example, the thalamus. So these various structures that are all right next to each other. So those are part of the thalamus isn't, but the basal ganglia, which is the central structure in the brain that, again, controls motor movement, does a lot of reward processing. Now on this right panel, if you cut the brain according to the red line on the left panel, you'll get something like what we see here on the right. And there, we can again see parts of the basal ganglia. So for example, again, we see the caudate and what's called the putamen, and we're not going to really delve into what each of these individual parts does so much. But those are part of the basal ganglia. This gives you a feel for where in the brain these things are located. You also have the amygdala right here, which again is part of the limbic system. So this is what these things look like in real life. You can also see here the distinction between, for example, the gray matter that I was talking about on the outer surface of the brain and the white matter, which is this lighter color in here. And as I mentioned, the gray matter seems to be where the action is for information processing. And that's why the brain surface is so convoluted, so wrinkled. It's to increase the surface area so you can get more gray matter for the same volume. The white matter, again, if we recall from a couple lectures ago, the white matter is white because of the myelin. So those are myelinated axons. And really what the white matter is it's moving information long distances. So from the occipital lobe to the frontal lobe. So you have a group of axons that are transmitting the information a long distance relative to the size of the brain. And so that's what the white matter does. The white matter is really a conduit for bunches of axons to get from one part of the brain to another. Okay, so now we saw this diagram last time as a way of listing the important neurotransmitters in the brain, but now we sort of have our geographic coordinates. So now we can understand a little bit more about this diagram. So obviously here we have, again, the person's nose would be on the left side of each of these brains. The top of their head would be at the top of the diagram. And what we can see is that these neurotransmitters, the neuromodulators, acetylcholine, dopamine, norepinephrine, serotonin, all originate around the middle of the brain, what's called the midbrain, and then send out axons, which we call projections, throughout the cortex, throughout the larger portion of the brain, the outside portion. And so we have these neuromodulator systems that begin in the middle of the brain and then send axons out throughout the rest of the brain. And there's relatively few of these neurons relative to the rest of the brain, which doesn't use the neuromodulators, it uses things like glutamate and GABA. So these neuromodulators project throughout the brain, but all originate pretty much in the same general area. Sort of like acetylcholine, it's in the basal forebrain. For the dopamine system, it is the substantia nigra and what's called the ventral tegmental area. These areas will come up again and again, so I'm not going to belabor them here. Norepinephrine comes out of what's called the locus ceruleus, which is just Latin for the blue place, because when they stain the brain, that particular part of the brain seemed blue. Serotonin comes out of what are called the raphe nuclei. But the point is that all these things originate in the middle of the brain and then go out from there. And that's true for these neuromodulators. Now, things like glutamate and GABA, which if you remember, glutamate is the main excitatory neurotransmitter, and GABA is the main inhibitory one. These things occur throughout. They're not localized in the same way that the acetylcholine or dopamine or other systems have this very specific location where the neurons begin, where the cell bodies are. Instead, glutamate and GABA, that's a lot of neurons, and it's all throughout the brain. So they're not localized in the same way. So that's the brain, sort of in a nutshell. How do we refer to different parts of the brain? So we have different systems in the central, peripheral nervous system, different lobes of the brain, different organs on the sort of the inner portion of the brain. How do we measure all this? So how do we know what we know about the brain? Well, we measure it. And so there are methods that can examine the brain structure. So how neurons are arranged, how axons go from one place to another. And of course, for whole brain techniques, the resolution is usually such that we can say gray matter is here, white matter is here. We're not looking at individual neurons, but we can localize the occipital lobe, the frontal lobe, the basal ganglia. And so we can look at structure, so different parts of the brain. But we can also look at function, that is brain activity. So which areas of the brain are active when we engage in a certain activity or when we receive a certain stimulus? How do we know what parts of the brain do what? Well, it's through measuring brain function. And so an experiment can look at metabolic activity, which groups of neurons are using up sugar and oxygen. Can look at electrical activity. Of course, action potentials are electrical impulses. So if you're measuring changes in voltage or current in part of the brain, if that area of the brain is active, the more action potentials it fires, the more electrical activity you'll see in that part of the brain. And of course, you can also look at chemical activity. So if I can detect a certain neurotransmitter, say dopamine, then, provided that dopamine neurons are present in the area I'm measuring, then if I can see changes in dopamine over time, I can infer that the dopamine system is responding to whatever stimulus or whatever activity the person is engaged in. So we can look at all these things. Also, methods can observe, so they can look at brain structure, they can look at brain function, they can also manipulate. We're not going to talk about that as much because it's not as relevant for most drug-related research. But there are methods that can interrupt brain activity. And so if you're interested in what a particular part of the brain does, you can try and determine what it does by shutting down that part of the brain temporarily. Or in certain animal populations, even permanently, you can lesion part of the brain and see what functions the animal loses. But of course, not every change in the brain, things like lesions, are intentional or experimental. So the fact is that repeated drug use can change the brain. So it can change the chemistry of individual neurons, which receptors the neurons express, what their pattern of activity is like. But of course, it can also affect the brain on a larger scale. Certain drugs, for example, are toxic to the serotonin system. And so you can lose those serotonergic neurons, and that can be observed with one of these measurements of brain structure or function in the serotonin system. Okay, so some of the earliest techniques for looking at the brain looked at brain structure. And one of the most common techniques is what's called computerized axial tomography, abbreviated CAT. So it's a CAT scan, if you've heard of that. It's also more commonly now called a CT scan for just computerized tomography. Same thing. And what this does is generate a 3D image. You can look at one slice at a time. And the images look like this. And so here you can see a large lesion or tumor in this portion of the brain. And so it's a structural technique. It looks at the brain's structure. And what a CT or a CAT scan really is it's a 3D X-ray. So it's the same technology that you get for X-raying your leg or your hands or whatever. And it's an old technology. It's been around for over 100 years. But what makes CAT or CT scan novel is that it does it in 3 dimensions. And it does it by taking an X-ray image and then rotating a little bit, taking another one, rotating a little bit, and so on, until you get a 3D reconstruction of the body. Another structural technique is what's called magnetic resonance imaging, or MRI, and many of us have probably had an MRI before. And this generates a similar structural image. It looks similar to a CAT scan, but MRIs could actually be sensitive to different properties of the body part in question. So it can be sensitive to the density in terms of molecules. So is it mainly water? Is it mainly fat? As we'll see, MRI can also be used functionally. But a structural MRI works by subjecting the body to a large magnetic field. And we're not going to go into the physics of it, but basically your body is full of protons, and when you put them in a magnetic field, they tend to align with the field, and then you can determine what sort of molecules are present by how those protons behave in the presence of the magnetic field. But again, it's slightly different from a CAT scan. It also has a certain advantage and disadvantage. It takes longer than a CAT scan. CAT scans are very quick. If you come into the emergency room, they'll often run a CT scan because it can be done fast, but they won't run an MRI because it's time consuming. The disadvantage is that a CAT scan depends on x-rays. So you can only have so many CAT scans in a year before you've reached your maximum dosage for radiation. As I mentioned, there's also functional imaging. So functional techniques looking at brain activity. One of these is called positron emission tomography, or PET. It's a PET scan, probably heard of that. And the way this works is you inject a radioactive substance into the body. Say, for example, you modify glucose, sugar, so that it's radioactive. And then it gets transported into the brain, so the brain can use it as energy. And depending on which parts of the brain are active, those parts of the brain will take up more of that glucose. And so they'll become more radioactive in effect. And what that radioactivity does is it shoots off these little subatomic particles called positrons, and then you're in this large circular machine, not dissimilar from an MRI machine, but this machine detects those positrons. And so you end up with a picture of which parts of the brain are active. There's also functional magnetic resonance imaging. The MRI part, of course, is what we saw on the last slide. The F part is new, functional. And this uses the same machine, but looks at a slightly different phenomenon. So what an fMRI does is it looks effectively at blood flow. Again, we're not going to get into the physics, but if an area of the brain is more metabolically active, then it's going to need more blood, because blood is what carries glucose and oxygen to brain cells. So if it's more active, it's firing more action potentials. If there's more synaptic transmission going on, it's going to need more glucose, it's going to need more oxygen, so it's going to need more blood. And so we can measure that change in blood flow over time based on what the person is doing or what stimulus they're receiving. So those are the two main functional techniques. Again, they have advantages and disadvantages. PET is very sensitive, but it's slow. You can only take about a picture a minute. fMRI, not as sensitive, but it's fast. You can take a picture every 2 seconds or so. So those are the trade-offs. Nowadays, fMRI is more common than PET for looking at brain activity, but for a time, PET was very popular. And of course, PET is used for other therapeutic applications as well. Okay, but not every technique is large scale. Not every technique looks at the whole brain at once. There's a group of techniques, really, called electrophysiology. And this is where you can take an electrode and insert it into a single neuron. So you can measure electrical activity in that neuron. And what you're really measuring, of course, are action potentials, which we've already talked about last week as being electrical impulses. So they're waves of ions that travel down the axon. And every time one of those action potentials occurs, the electrode can detect it. So it's measuring changes in voltage. So you can look at the activity of individual neurons and see what an individual neuron responds to, what it sort of cares about, so to speak. And this is the way that a lot of the brain was mapped out. So we know that a certain part of the brain is responsible for moving the hand. If we record from that area, And every time the person moves their hand, we get a whole bunch of action potentials in the neuron we're recording from. You can also actually record from individual neurotransmitter systems. So I mentioned dopamine as a way of looking at chemical activity. So you can actually stick an electrode in the brain, and again, as long as you're in a dopaminergic area, you can measure the level of dopamine second by second, and look at how dopamine changes when, for example, a drug is given to the animal, or if a reward is given that isn't a drug, or something unexpected happens. You can look at all sorts of stimuli and look at dopaminergic activity as a function of the stimulus. There's also microscopy, which is just a way, it comes from the root microscope, so looking at neurons through a microscope. And this will let you look at the detailed structure of the neuron. So you can look at the dendrites, the little spines that come off the dendrites to make synapses. So you can look at synapses, whether new synapses are forming or whether they're being deleted. Synapses form and go away all the time. But for example, you can look at dendritic spine formation as a function of whether a drug has been administered. Okay, so that's it for techniques. Back to synaptic transmission for a minute, because we're going to lead into what drugs do. So we've been talking about the nervous system and really primarily the brain this whole time. But what do drugs do, and how do they work? Why do they work? So if we recall, for the presynaptic neuron, an action potential comes down the axon into the axon terminal, and that triggers a set of events in the presynaptic neuron. The calcium comes in, synaptic vesicles attach to the membrane, and open up and release neurotransmitter. So that's the second step. As neurotransmitter gets released into the synaptic cleft, that is the space between the pre- and postsynaptic neurons. And so these receptors on the postsynaptic neuron, once the neurotransmitter diffuses across the cleft, they latch on, the neurotransmitter molecules latch onto the receptors on the postsynaptic neuron, and the receptors either open up if they're ion channels or trigger some metabolic cascade if they're metabotropic receptors. And then, of course, there are events in the postsynaptic neuron, like letting in ions and having these chemical cascades. And so that's how the presynaptic neuron has an effect on the postsynaptic one. So where do drugs come into all this? So we've already talked about the normal process of synaptic transmission, and drugs can really affect any part of this process. They can affect neurons in general. So one of the things that alcohol does is it affects the membranes of every neuron all over the neuron. So it's a very general effect. And it's thought that anesthesia works in much the same way. Anesthesia isn't very well understood, but it can affect neurons all over. Or a drug can affect the synthesis of a particular neurotransmitter. And so if the neurotransmitter gets made in abundance or if production ceases, obviously that'll affect the ability of the neuron to release neurotransmitter into the cleft. It can also prevent neurotransmitter from being stored in those synaptic vesicles. So if it inhibits those synaptic vesicles, then they can't be released and you can't get synaptic transmission. It can affect the release of neurotransmitter. So maybe it gets stored in synaptic vesicles just fine, but you somehow inhibit the vesicles fusing with the membrane and releasing those neurotransmitters. On the postsynaptic end of things, and this one is fairly common, you can have a drug that binds to the receptor. So if the neurotransmitter normally binds to a receptor, but it can't get to the receptor because a drug is already attached, well, that's going to affect the function of that neurotransmitter. Or if it actually mimics the neurotransmitter, then that receptor is going to act like that neurotransmitter is out there all the time, even if the presynaptic neuron isn't firing, isn't releasing neurotransmitter. You also have the stage of synaptic transmission once the neurotransmitter is diffused across the cleft. As we saw about last week, that neurotransmitter eventually gets pulled back into the presynaptic neuron or destroyed. And so if you prevent what's called reuptake, that is, the sort of vacuuming up of a neurotransmitter back into the presynaptic neuron, if you keep a reuptake from happening, then all that neurotransmitter is going to stay out in the synaptic cleft, and it's going to be able to be more effective. It's going to enhance the effect of the neurotransmitter because it keeps on bouncing around in the synaptic cleft and never gets sucked back up into the presynaptic neuron. We also mentioned the possibility that enzymes in the synaptic cleft can break down neurotransmitter. If you interfere with those enzymes, then again, you're going to have more neurotransmitter out there in the cleft, and what's going to end up happening is you'll have neurotransmitter attaching to receptors more frequently because there's more neurotransmitter bouncing around out there. It's not being chewed up. So there are all these ways for drugs to work, and there are different types of drugs and different ways of describing them. So one of the big distinctions is made between agonists and antagonists. So antagonist, just like an antagonist in a story, is something that works against the normal action of, in this case, a neurotransmitter. So if a neurotransmitter normally activates a receptor, an antagonist would, in one way or another, it's not specific to how, but an antagonist inhibits the action of that neurotransmitter. You can do it in a variety of ways, but an antagonist diminishes the activity of that neurotransmitter. An agonist is the opposite. It either mimics or in some way enhances the action of the neurotransmitter. So those are two opposite concepts. You also have direct and indirect action. So you can have a direct antagonist or an indirect antagonist. And all this really means is a direct antagonist links directly to the receptor, whereas an indirect one disrupts or in some way affects some other aspect of synaptic transmission. So again, if it affects reuptake or affects the enzymes that break down neurotransmitters, those would be indirect forms of action. Whereas if it binds to a receptor and keeps that receptor from working, that would be a direct antagonist. And you can mix and match these terms. You can have direct antagonists or indirect ones, direct agonists and indirect ones. And we'll talk about all these things as we talk about individual drugs. So how does all this work? We talked about receptors a little bit. One thing to keep in mind is that receptors are actually very complicated. We talked about a receptor response to a neurotransmitter, and we talked about ionotropic and metabotropic receptors. But the fact is that, certainly in the case of the NMDA receptor that we see here, and what that means is not important. But it's not just glutamate in this case that's binding this receptor. All sorts of stuff can bind to it. And the reason this is it allows modification of the action of that receptor. NMDA receptors are very important for memory and for learning. And so to learn something, you have to have a whole bunch of things going on at once. And so you have this complex receptor that can respond to a variety of chemicals out there in the synapse. Now these happen to be ionotropic receptors, which means that they open up and let ions in as their mode of action. You also have metabotropic receptors, which we talked about last time. And these, again, trigger those chemical cascades. But neurotransmitters can connect to any of these things, but also drugs can too. So these are just molecules, and drugs, and these are direct agonists and antagonists because we're talking about receptors. Drugs can connect to these receptors and make them permanently open, so they're active all the time, permanently inactivate them, so they're closed all the time and no neurotransmitter can bind to and open the receptor. And so drugs can interact with these receptors because these receptors have to be complex. And oftentimes, especially naturally occurring drugs, evolved as a response to the presence of these receptors. So various molecules have been evolved by plants as defense mechanisms. So they evolved some molecule that if an organism eats the plant, that molecule is now floating around, binds to some receptor. Now that receptor doesn't work the way it used to. So these are things like poisons. And many drugs started off as poisons, and they're just drugs because you take them in smaller dosages. So what's the point? The point is that receptors can have a lot of what are called endogenous ligands. Endogenous means made within the body, native to the body. The ligand part just means something that can bind to a receptor. So it's something that naturally occurs within the body to bind to these receptors. That opens up an opportunity for exogenous substances, that is, substances that aren't native to the body, to imitate the endogenous ones. So that's the way a lot of drugs work, is that drugs imitate a molecule that already exists in the body and then works slightly differently, or you take a lot of the drug and that concentration is so different from what the normal level of the substance is that you have an adverse effect on neural function. Okay, so now we understand a little bit about how drugs have their effects, at least at the neural level. But of course, we care about describing the effect of drugs bigger than the neural level. So we want to understand their effects on behavior. Again, this is a course on drugs and behavior. Human behavior is what, in the end, we really care about the most. But we need to understand how they affect neurons in order to fully understand the effects on the nervous system and behavior down the line. So one of the easiest ways to quantify what a drug does is in terms of what's called the dose-response curve. And this is shown in the diagram on the right. So on the x-axis, the horizontal axis, you have the dosage, in this case of alcohol. On the y-axis, you have the percentage of people that are affected. So what this does is it acknowledges the fact that everyone is not the same, whether by virtue of age or body weight or gender or what have you. Everyone responds to a drug slightly differently. So it might not take as much of a drug for one person as it does for another to have the same behavioral effect. And so a dose-response curve is a way of illustrating that. So you see these curves, and it shows that a low dosage of alcohol affects a small percentage of the population, and a larger dosage affects eventually everyone. If you bump up the dosage enough, eventually it's going to have an effect on everyone. But it's a way of looking at the population of people or animals and saying, what's the population like in terms of how it responds to the drug? Another concept is the effective dose, which is how much the drug is, what's the minimum amount of drug needed to have an effect on behavior. And the ED50, which is ED is for effective dose, 50 is for 50%. What's the required dosage to have a behavioral effect on half a population? So on average, how much do you need to affect behavior? The dark side of that is the lethal dose, and its analog, the LD50. So the lethal dose, as the name suggests, is how much of the drug it takes to kill the individual. And the LD50 is the lethal dose for 50% of the population. So how much of the drug is required to kill 50% of the individuals in a group? Again, there's some variance in a group. So on average, how much of the drug is a lethal dosage? Now you can combine these two measures into what's called a therapeutic index. And all that is it's the ratio of the LD50 to the ED50. So how much is required to kill the individual compared to how much is required to be effective. Now that number is always going to be greater than 1 because it always requires more of the drug to kill the person than to affect the person in some way. But of course, if it's just above 1, what does that mean? It means that it only requires slightly more to kill the individual than to affect their behavior. So that's not much of a margin for error if we're trying to figure out our dosage. If we have to have a certain amount to have any effect at all, and only slightly more to be lethal, that's a dangerous drug to work with. Of course, if the ratio is very big, it means that you only need a little bit, relatively speaking, to have an effect on behavior, but you need a whole lot to kill the person or the animal. So that's good. So you have a lot of range in there in which you're safe. And speaking of safe, another measure is what's called the safety margin, which is really just the difference between how much the ED50, how much is required to, on average, have an effect, and what's the dosage I need to kill anyone at all. So not the LD50, it's like the LD1, so to speak. So that, again, this is a measure of the range we have to work with. We don't want to, if we're manufacturing a pill, we don't want to put in an amount of the drug that is able to kill anyone, much less 50% of people. So really, if we're making a drug, our concern is not the therapeutic index so much as the safety margin. So we don't want to kill anyone, So we can have a dosage less than the lethal dose for anyone, but at least the effective dose for half of individuals. So that's a look at how we quantify the effects on behavior. But of course, the effects aren't just on or off, they happen over time. So we have to care about the time course of these effects. And there are two important points here. This is a diagram from your book. One of the things it demonstrates is that there isn't just a proportional relationship between the concentration of the drug in the blood and its effects on behavior. It can be what's called nonlinear, which is to say you can have some in your blood and it doesn't affect you at all, and you put more drug into the bloodstream, and now you have a decidedly measurable effect. So you can have this sort of quick increase in the behavioral effect with just a small increase in dosage. So it's not just that you can estimate the effects on behavior by looking at the concentration in the blood. And this works for both as the drug becomes more concentrated in the bloodstream, as the drug starts to take effect, and as it starts to go away, as the body metabolizes it or gets rid of it. So we can have these nonlinear effects over time. The other important point is that what this means is that you can't just take twice as much and expect to have twice the effect. If you take a certain pain reliever and you decide it's not having the effect you want, you can't just go and take twice as much and it'll double the effectiveness, because that's not how it works. You might have reached the maximum effectiveness, and then adding more drug into the bloodstream is really not doing anything for the pain, but it does have an amplifying effect on side effects, which is not what you want. So those are the two take-home lessons, is the effects aren't necessarily just a function of concentration in the blood, and you can't just double the concentration in the blood and expect double the behavioral effect desired. Okay, so that's it for this lecture. Next time we will talk about drugs in the brain again, but looking at a larger scale. So we're talk about the brain as a whole and behavior. And so the first thing we want to do is try to categorize drugs so it's a little bit more manageable. So on what basis do we categorize drugs? Which categories are more useful than others? We'll talk about that next time. We'll also talk about methods of administration. How do we get drugs into the body? There are various methods. What are the implications of those various methods for the brain and the rest of the body? And then what are the longer term effects of drug use? After repeated use, what happens to neurons, the brain, the body? Okay, that is it for this lecture. I will see you all for the next one.